Hello all, I am Mr. Prabakar, Assistant Professor. I am here today to talk about the hypersensitivity reactions. The learning objectives for this session would be, at the end of the session, you will be able to define and classify hypersensitivity reactions, differentiate between the 4 types of hypersensitivity reactions, describe the immunological mechanisms involved in each type of hypersensitivity reactions, list the clinical signs and symptoms associated with each type of hypersensitivity reactions. Let's look at the definition. Hypersensitivity reaction is defined as an exaggerated, inappropriate state of normal immune response with onset of adverse effects. It is a process or a condition at which the body's immune response functions beyond the required amount. As a result, there might be injurious consequences to the sensitized host following a subsequent contact with the specific antigen. The classification of hypersensitivity reactions can be categorized into two. First one, based on Gell and Coombs classification, they are divided into type 1 anaphylactic reactions, type 2 cytotoxic or cytolytic reactions, type 3 immune complex mediated reactions, and type 4 delayed hypersensitivity reactions. Looking at the classification, the second type is based on the time taken for the reaction to occur in the body. We can categorize them into immediate and delayed type of hypersensitivity reactions. The major difference is that the time to manifest these happens from minutes to hours in immediate, whereas in delayed it takes about days. Type 1, 2 and 3 come under the immediate type hypersensitivity reactions, whereas type 4 comes under the delayed type hypersensitivity reaction. The major mediators for type 1, 2 and 3 are antibodies, whereas that of delayed is the T lymphocytes. The route of sensitization could be any route for the immediate type of hypersensitivity reaction, whereas for the delayed type it will be the intradermal. Now, the hypersensitivity reaction can be transferable in immediate type through serum, whereas it is not possible in case of the delayed. The desensitization process is easy in case of immediate hypersensitivity reaction, whereas in case of the delayed hypersensitivity reaction it is difficult, however long lasting. Moving on to the first type 1 hypersensitivity reaction, it is also called as anaphylactic or atopic reactions. They are defined as a state of rapidly developing or anaphylactic type of immune response upon an exposure to an allergen. So, a previous sensitization to the allergen is necessary in order to develop this hypersensitivity reaction. The reaction occurs within minutes after exposure to the antigen, and the antibody depending on the type 1 hypersensitivity reaction is majorly IgE. Etiology of type 1 hypersensitivity reaction, the immediate reaction following an allergen sensitization could be: number 1, based on genetic basis, the allergic reactions may be linked to the hereditary inheritance like asthma or hay fever, whereas second, environmental pollutants such as the pollen or poison ivy can also cause type 1 hypersensitivity reaction. The third common factor is a concomitant factor, which is nothing but the simultaneous occurrence of viral infections could sometimes lead to type 1 hypersensitivity reaction. The pathogenesis of type 1 hypersensitivity reaction includes cells such as B lymphocytes, plasma cells, mast cells, basophils, neutrophils and eosinophils. The host has to get sensitized with an allergen or the antigen first, and in response to this the B lymphocytes gets activated, which differentiates into plasma cells producing IgE, and these IgE bind to the mast cells and basophils and are now fully sensitized to this particular allergen. Upon re-exposure to the same allergen, the second interaction causes the release of histamine granules from this sensitized basophils and mast cells, leading to type 1 hypersensitivity reaction. Looking at the pictorial representation of my explanation, you can see here the first exposure allergen comes in contact with the antigen presenting cells. The antigen presenting cell are then activate the T helper cells and B lymphocytes, which ultimately develop into plasma cells producing IgE. The IgEs go and bind to the mast cells or the basophils and sensitize the mast cells and the basophils. Upon the re-exposure, the allergen comes in contact with this sensitized mast cell or basophil, releasing the histamine chemicals or chemokines, causing the type 1 hypersensitivity reactions. There are primary and secondary mediators for the type 1 hypersensitivity reactions. The primary mediators are basically histamine, protease, eosinophil chemotactic factors, neutrophil chemotactic factors and heparin, whereas the secondary mediators are platelet activating factors, leukotrienes, prostaglandins and bradykinin. The effect of these mediators are such as increased vascular permeability, which causes smooth muscle contraction, early vasoconstrictions followed by vasodilation. In some cases it can lead to shock, increased nasal and lacrimal secretion and increased migration of eosinophil and neutrophil. Examples of type 1 reactions: systemic anaphylaxis. Such examples are antitetanus serum, which causes type 1 hypersensitivity reactions. Drug administrations can also cause type 1 hypersensitivity reactions. Bee or wasp sting can also cause type 1 hypersensitivity reaction. The clinical features of type 1 are generally itching, erythema, some cases respiratory bronchial contraction, and in rare cases diarrhea also can be seen. Local anaphylaxis, we have hay fever, also called as allergic rhinitis, bronchial asthma, food allergy in some cases such as fish, milk, eggs, etc. can also cause type 1 hypersensitivity reactions. Cutaneous urticaria, wheal and flare reaction will be seen in the local anaphylaxis reactions. Type 2 hypersensitivity reaction, also called as cytotoxic or cytolytic reactions. As the name suggests, this reaction is majorly by humoral antibodies that attack the cell surface antigen, which results into cell lysis of the targeted cell. This reaction roughly takes about 15 to 30 minutes after the exposure to the antigen to be seen in the body. Etiology of type 2 reaction basically involves complement system, tissue macrophages, platelets, natural killer cells, neutrophils and eosinophils, while main antibodies involved are IgG and IgM. Type 2 is tissue specific and the reaction occurs after the antibodies bind to the tissue specific antigen, and only then will you be able to see the type 2 reactions in the body. The pathogenesis of type 2 reactions, you can see the target cell contains the surface antigens. The surface antigen will be recognized by the antibody binds to the surface antigen and will activate the T lymphocytes or the natural killer cells, which in turn activates the complements leading to membrane attack complex or alternatively leading to opsonization by phagocytosis. The end result of this would be the destruction of the target cell. Examples of type 2: autoimmune hemolytic anemia, transfusion reaction, erythroblastosis fetalis, drug-induced cytotoxic antibodies. These are all type 2 reactions that are associated with the blood cells. When you look at the tissue components, the examples are cellular injury due to autoantibodies, Graves' disease, myasthenia gravis and male sterility. These are some of the examples. Type 3 hypersensitivity reaction is also called as immune complex mediated reaction, also known as Arthus reaction. This reaction majorly depends on the antigen antibody complex, also called as the immune complex, when they get deposited on tissues, and improper removal of this immune complex can lead to the activation of complement system and inflammatory reaction, which leads to cellular injury. The onset for this reaction to happen roughly takes about 6 hours after the exposure of the antigen. The etiology is that type 3 is not tissue specific and occurs when the body fails to remove the immune complex. Formation of immune complex, antigen antibody complex, is a common way of how the antigen is removed from the body or destroyed in the body. If this antigen antibody complex, when formed, is not removed from the body, then it can deposit on the tissue, causing type 3 hypersensitivity reaction. There are 3 types. Number 1, persistent of low grade microbial infection, where the antigen will lead to a weak antibody production. This weak antibody results as a loss or reduced affinity to the antigen. Hence, antigen antibody complex will be formed, but not strong enough to remove or destroy the antigen. Hence, more and more immune complex will be formed, and these immune complex can lead to type 3 hypersensitivity reaction. Secondly, extrinsic environmental antigens, exogenous antigens when inhaled in the lungs can also lead to type 3 hypersensitivity reaction, where the antigen will act as the weak antigen and will not be removed off, resulting into antigen antibody complex formation. Autoimmune processes where autoantibodies are produced against self antigen can also lead to type 3 hypersensitivity reaction. Example, autoantibodies to DNA or myelin sheath of the neurons can lead to the destruction of these, resulting into a type 3 reaction. The pathogenesis is that the antigen comes in contact with the B lymphocyte. The B lymphocyte identifies the antigens and get activated, resulting into plasma cell and antibody production. Usually the antibodies go and bind to the antigen and get destroyed either through phagocytosis or other reactions. Here the immune complex formed is not removed from the body and they do not destroy the antigen. Hence, the immune complex attach themselves to the tissues, leading to activation of complement as well as the neutrophil, which will in turn result into tissue damage. This results into type 3 hypersensitivity reaction. Examples of type 3 hypersensitivity reaction: immune complex glomerulonephritis, Goodpasture syndrome, systemic lupus erythematosus, rheumatoid arthritis, farmer's lungs, drug-induced vasculitis. Moving on to the fourth hypersensitivity reaction, it is also called as delayed type hypersensitivity reaction. It is also called as cell mediated hypersensitivity reaction. Cell mediated indicates that this particular reaction results into tissue injury, which on contrary to the type 1, 2 and 3 antibodies are not involved. Here the cell involved is a sensitized T lymphocyte. The reaction takes about 24 hours after the exposure to the antigen and can persist up to 14 days in the individual. The etiology is that type 4 reactions involves mast cells, basophils, macrophages and CD8 plus T cells. These cells, when the antigen is presented by the antigen presenting cells, will interact with the T lymphocyte. The T lymphocytes get sensitized and activated, which will result into destruction of this particular antigen. However, simultaneously it will also produce cytokines which will activate macrophages. The activated macrophage can go and form granuloma around the site of injury or inflammation and can lead to type 4 hypersensitivity reaction. Here majorly the T lymphocytes are being sensitized and overactivation of the T lymphocyte results into type 4 hypersensitivity reactions. The examples of type 4 reactions are mycobacterial infections such as tuberculosis infection, virally infected cells, malignant cells in the body, organ transplant, graft versus host reaction and transplant reactions all follow under the type 4 delayed hypersensitivity reaction. Moving on to the summary of my session. The mediator for type 1 is majorly IgE, whereas that of type 2 is IgG or IgM. Type 3 immune reactions will have antigen antibody complex as the mediator, whereas type 4 will have T lymphocytes. The antigen that is resulting into these reactions, you can see that soluble antigens are the ones which induce type 1 and type 3 reactions, whereas type 2 is majorly induced by cell bound antigen, whereas type 4 can be caused by both soluble as well as cell bound antigens. In type 1, the allergen results into IgE production which activates the mast cells leading to the reaction. Type 2, IgG and IgM antibodies will activate complement resulting into destruction of the cells, whereas in type 3 the antigen antibody complex again activates the complement which results into inflammations, and in type 4 the antigen presenting cells activate the T lymphocyte which in turn releases inflammatory cytokines leading to the reactions. The time taken is roughly about minutes to hours in case of type 1. Type 2 and type 3 it roughly takes about hours to some days, whereas type 4 usually occurs 24 hours after the exposure to the allergen, and it is a delayed type which results in more than 14 days of time. Some examples of type 1 are anaphylaxis and hay fever, type 2 hemolytic disease of the newborn, type 3 rheumatoid arthritis, post-streptococcal glomerulonephritis, and type 4 is contact dermatitis and graft rejection, etc. So, to summarize, these are the 4 types of hypersensitivity reactions. These are my references. Thank you.